circulatory system, the complete series. Previously already we have discussed the surface protein of that RBC. So this is the RBC. In that RBC for ABO blood group system, we already discussed the band 3 protein. So band 3 are like a transmembrane proteins are there in the surface of the RBC, and with that band 3 protein, ABO antigens are get bind with that. Also, I'll go through that the exactly what are there like H antigens are there in that system. So and also we just checking the other like MN systems is there. MN system also a blood group system, and LW, Duffy, Indian, Lutheran, YT, and RH blood group system. It's linked with that ABO blood group during transfusion of the blood. Kale and Cromer, knobs, and Diago. So these are different kinds of proteins present on that surface of the RBC. That's why the blood group gets changed. Why it is essential? During transfusion of the blood, it's necessary to match that blood group. Otherwise, the different type of antigen will come in the body, and body have different kind of antibody, and then interact happen, then blood agglutination happen. With that, so for ABO system, A allele, B allele, and AB allele, both the expression, that is the co-expression of that allele is there. And in O, nothing will express. They are also RH positive and RH negative. For that, we get the 8 types of blood group: A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, and O negative. So in Indian population, most of them are in the B positive blood group, but it's like kind of prediction on that. So along with the AB positive and like AB negative and O negative, kind of rare blood group, but still we can get. So these are the antibody against this antigen. These are the anti-A and anti-B. These are like IgM antibodies. These are present in the blood. Let's go for the details of the ABO antigens. Like for this antigen, it's one of the essential antigen is the H antigen, and these all coded by the chromosome 19 that have the 3 exons, and that give the 5 kb of total gene, and 5 kb of the total gene, and that can express the flucosyl transferase. In the H antigen, you can see the structure. The glucose is there, and then galactose is there, and in acetyl glucosamine is there, and then galactose is there. So in them, the alpha-L fucose is binding with that galactose in that chain. So these are the common, and with that gene, like in 261 position, the delete of that exon 6. That's why due to deletion of that one nucleotide, like it's a kind of frame shift mutation. Due to that they don't have the like GalNAc transferase of that. That's why it's kind of H antigen without binding anything. And for A antigen, the binding of the N-acetyl galactosamine, how it is bind? The A allele, the in chromosome 9, that's the in ABO locus, chromosome 9, A allele encodes the GalNAc transferase. In this transferase enzyme helps to bind in their N-acetyl galactosamine in the galactose position, in the galactose. So that gives the special feature. And if A antigen is there, so like antibody, their anti-A antibody will develop here. So like these are the anti-antibody, and like that the in beta D galactose, also alpha D beta galactose is bind with that galactose position. So that is known as the B antigen. So in AB blood group, both the type of A antigen, these allele also express, and these are also expressed. So for AB blood group, both the kind of genes are get expressed. So that is called the co-expression of that allele, and that give the AB. And if H is the risk factor is also present there, that will be like the AB positive. Let's try, and hope you got the understand. Now we are coming to how experiment is done and how check the blood group is there. So like we take a slide and take 4 drops of the blood. One always will be the control, and it will be like no agglutination on that in control, because we have to confirm that the experiment is right. So in control, the no agglutination is there, and in one we'll give the anti-A, in one section we'll give the anti-B, in one section we'll give the anti-D. Anti-A if the agglutination is not there, A, B, but D is there, that means the in O positive. They like anti-A, uh, because in O positive it means there is no kind of antigen. So that means anti-A cannot bind, anti-B cannot bind, but RH positive there and their antibody is binding with that. That means it's a O positive. In first case, it's a O positive. Like that, you go for the AB positive. Like A is there also, B is antigen is there, and D antigen is there, and along with antibody we have provided. That means the antigen antibody reaction happen. That means the agglutination happen, and we confirm that the AB positive. And if in every section agglutination is there, that means the experiment is not valid. Okay, let's go for two special type of blood group. One is the Bombay blood group. H is no H antigen. This is the Bombay blood group, and small type of populations are already, but and along with some other type of blood group system like A main S system blood groups are there. These are kind of rare kind of things, and all are depends on different kinds of antigens, and antigens are there. So hope that this class you can understand this class. It's very easy. ABO blood group system, and you have to, for exam purpose, you have to remind the which chromosomes like fucosyl transferase is encoded by the chromosome 19, and the ABO locus present in the chromosome 9, and GalNAc transferase enzyme and their function, and the antigen antibody and IgM, and along with some surface protein band 3 and RH, where they are present and how blood groups are essential. Hope that this class can benefit it.